When a virus replicates, mutation in its genetic sequence occurs, which may alter some characteristics of the virus. If the altered virus mutated significantly enough from the original virus, and gained survival advantages through these mutations, it may spread rapidly in a host population, becoming a new variant. The SARS-CoV-2 alpha variant is one of the earliest mutated strains from the original SARS-CoV-2. It includes the B.1.1.7 strain, and with a much lower prevalence, the B.1.1.7 E484K strain. The alpha variant was first found in September 2020 in the United Kingdom. As of July 2021, it has spread to at least 157 countries with a cumulative prevalence worldwide of 44%. While, in the United Kingdom, it has an even higher cumulative prevalence at 52%. On 18 December 2020, the alpha variant was classified as a variant of concern. What are the key mutations acquired by the alpha variant that makes it a variant of concern? From the genetic sequencing of many virus samples from infected individuals, it was discovered that the alpha variant has accumulated over 18 mutations with about 8 mutations on the spike protein. This information is of great significance as the spike protein is involved in both the transmissibility of the virus and the immune response by the host against the virus. This is the general structure of the SARS-CoV-2, and the red protrusions on the surface of the virus are the spike proteins. For the SARS-CoV-2 to replicate in host cells, it must first use its receptor binding domain on the spike protein for attachment to the cells. In addition, the cell surface protease TMPRSS2 facilitates the cutting of a part on the spike protein, called the furin cleavage site, allowing the virus to enter host cells for viral replication. Moreover, most antibodies generated through infection or vaccination are stimulated by the spike protein. In other words, the immune system of the host eliminates the SARS-CoV-2 by recognizing the spike protein. From the list of mutations on the spike protein, a few mutations have been extensively studied to understand their impact. The most notable example is the N501Y mutation, which is common in other variants of concern including beta and gamma variants. This mutation involves a substitution of the amino acid asparagine at the 501 position in the chain, for the amino acid tyrosine. N501Y mutation is the main cause of the increase in transmission rate of the virus, which led to spikes in COVID-19 cases. In addition, the alpha variant also has the D614G mutation, which substitutes the amino acid aspartic acid at the 614th position in the chain, to the amino acid glycine. This mutation is also common in other variants of concern including beta, gamma and delta variants. The single change in amino acid from both mutations slightly alters the structure of the spike protein, which increases the rate of virus binding to ACE2 receptors on host cells. Increase in binding allows viruses to enter the cell more easily, producing more viral particles, especially in the upper respiratory tract, and making it easier to transmit virus to other people, hence, increasing the rate of transmission. Other mutations have also been studied. For example, DEL 144-155 has been linked to viral resistance to antibody treatments. DEL 69/70, which involves the deletion of amino acid, has been suggested to cause diagnostic test failure, giving false negative results. In general, the mutated characteristics of the alpha variant include about 50% more easily spread among the community than the original SARS-CoV-2, increased potential in disease severity based on the rate of hospitalizations and fatalities. However, antibody treatments are still effective against the alpha variant. Besides that, there has been little to no reduction in efficacy of COVID-19 vaccines and immune protection from reinfection. Examples of vaccines that are still effective include Pfizer, AstraZeneca and Moderna. Do note that these examples are based on available studies so far. Vaccines not listed by no means signifies non-efficacy but rather due to limited available data. Therefore, this provides evidence that 2 doses of these vaccines are still effective against the alpha variant with more than 50% efficacy, which is the efficacy cutoff point set by the Food and Drug Administration. If you enjoyed this video, don't forget to like, share or subscribe to my channel. If you would like to learn more about other variants of concern, check out this series.